This video will explore various processes of evolutionary change. After watching the video, you should be able to apply the concepts of Hardy-Weinberg equilibrium and see how we can use that to make predictions on expected frequencies of alleles and of genotypes in non-evolving populations. You should also be able to explain various mechanisms of evolutionary change. So keep in mind, the topics we're focusing on here will be applied to populations of organisms. So whether it be fish, penguins, humans, any organisms for that matter, we're talking about populations of organisms of the same species that live in the same area at the same time. So let's begin by thinking about this basic question. We addressed this in a previous video. So what is evolution? Well, at the most basic level, we can think about evolution as a heritable or a genetically based change in the characteristics of populations over time, or simply a change in allele frequencies over time, meaning generation after generation after generation. So keep in mind, of course, these types of changes can lead to larger scale changes, such as the formation of new species. And that'll be the subject of a separate video. So if evolution essentially the changing of frequencies of alleles in populations over time, let's think about a population that is not evolving. If we have a population that's not evolving, then the frequencies of all alleles, right, and all of the chromosomes and so forth will remain unchanged generation after generation. So this basic concept was thought about by several individuals. These two individuals, Hardy and Weinberg, that typically get credit for this work. By the way, they were not working together. They were working totally separately, but they kind of came up with the same basic concept that we're discussing here. So Hardy and Weinberg figured, okay, if various assumptions are met in populations, then we can say those populations would be in equilibrium. In other words, they would not be evolving over time. So, what are some of the assumptions that would have to be met for a population to not be evolving? Well, first of all, you cannot have any natural selection, right? No phenotypes can be selected for. None can have an advantage over any other phenotypes. In addition, there can't be any genetic drift. By genetic drift, we'll come back to this in a minute. By genetic drift, we simply mean random changes in allele frequencies due to, again, just due to random events. We'll come back to that in a bit. You can also have no gene flow, meaning that gametes from your population have to remain in your population. You can't have mating with individuals from other populations. In addition, you can't have any mutations because, as we'll see, that's the ultimate source of new alleles in populations. And you have to have random mating. Now, these are some assumptions that, again, would have to be met for a population to not be evolving. So if you think about these conditions, they're simply not ever met in natural populations. There's always at least one, if not several of these conditions that are violated, right, in natural populations. So let's just explore how Hardy and Weinberg sort of described this concept of non-evolving populations using a little bit of math, but conceptually, let's think about the following. Think about a gene. It could be for, let's say, earlobe attachment. You've got two versions of a gene, let's say dominant and recessive. One codes for free earlobes, one codes for attached earlobes. So, but it doesn't matter what gene you're talking about. But if you took a population of organisms, in this case, let's stick with humans, you can imagine, again, if you've got a dominant and a recessive version of that particular gene, if you looked at all of the dominant alleles, let's just say capital B, you can use whatever letters you like, and all the recessive alleles, let's say lowercase b, if you add all of those alleles up in your population, right, that's going to equal 100%, right? Because if you just have two versions of this gene, whether you have 15 individuals or 1,500 individuals, right? You add them all up, all the dominant recessive, collectively, that's 100%, right, of those B alleles in your population. Okay, so think about that contextually. Then let's think about genotypes. You can think about individuals in your population as being either homozygous dominant, big B in this case, homozygous recessive, little b, or heterozygous, right? Big B, little b. Again, those are the only options, right? You're either one of these three genotypes. So again, collectively, add all these genotypes up in your hypothetical population, and again, that equals 100%, right? Again, because there are no other options except those three that are listed. Okay, so what Hardy and Weinberg did was say, well, let's sort of formalize some relationships here. Unfortunately, I guess you might say they decided to use P and Q to designate, let's just say, the dominant and recessive version of allele. You could use big B, little b if you want here. Same idea. Don't let the letters confuse you. So if you want to think of P as your dominant allele and Q as a recessive allele, perfectly fine. You can just do that if that makes more sense. But again, they said, okay, if you add dominant and recessive alleles, add all those up, that'll give you 100% or a frequency of 1, right? So you have to keep in mind, this represents the frequency of alleles, those two alleles, dominant and recessive. Okay, what we can then do is essentially just square that equation. And we end up getting something like this. P times P plus 2 times P times Q plus Q times Q, again, equals 100%, or 1. So what this represents is the frequency of genotypes. Homozygous dominant, homozygous recessive, and heterozygotes. Another way to write that equation, exact same thing, is P squared plus 2PQ plus Q squared, again, equals 1. So, and again, this gives us a way to work with allele frequencies, right, in this first equation, and genotype frequencies in this third equation. So those two equations are used very commonly to answer or address basic questions related to Hardy-Weinberg equilibrium. So I have this image up on the screen. Feel free to pause the video at any point if you'd like to kind of walk through the logic here. What this is essentially illustrating is the logic behind Hardy-Weinberg equilibrium. And essentially what it's depicting is, let's imagine a hypothetical population, 20 individuals, let's say they're diploids. So each individual has two alleles, of this A gene, could be dominant, could be recessive. So 20 individuals, you have a total of 40 alleles, right, in this population. And let's just say in this hypothetical population, to begin with, we have 28 of those 40 alleles, the dominant version, so capital A, and 12 are the recessive version, lowercase a, right? So that gives us an initial frequency for dominant allele of 0.7, recessive allele of 0.3. And what this shows is so now we have this pool of genes, right, in these individuals. So the rest of this figure shows, okay, if these individuals were to mate, and if we didn't violate any of the Hardy-Weinberg assumptions, what we would see is that in the next generation, sure enough, the frequency of our dominant allele, capital A, remains at 0.7, and our recessive allele, lowercase a, remains at 0.3. So again, feel free to pause this video if you'd like to sort of walk through the logic that's illustrated here. But let's take a look at a sample question to see how we can apply this concept. So there's the question. You have a remote mountain village, and 64% of the individuals can taste this chemical, PTC. And those individuals that can taste it, they have at least one dominant allele for this gene. So they could be homozygous dominant, or they could be heterozygotes, right? Both of those genotypes would be individuals that can taste PTC. And so then if this population conforms to the Hardy-Weinberg expectations, in other words, if no assumptions are being violated in this population, what percentage of the population is expected to be heterozygous for this trait? And they give you some options here. So again, don't forget our equations that we mentioned earlier, p plus q equals 1, and then this one here, p squared plus 2pq plus q squared equals 1. Let's take a minute and walk through how we would go about solving this question. Okay, so I'm just going to rewrite some of these relevant equations here. So p plus q equals 1, that's a frequency of alleles in the population. And then we said p squared plus 2pq plus q squared equals 1. And that allows us to work with genotypes in our population. So in our question, we're given that 64% of the individuals can taste it. We have no idea what their genotypes are. They could all be homozygous dominant. They could all be heterozygotes, or there could be a mixture, right, of homozygous dominants and heterozygotes. We don't know for sure. We just know that those individuals that can taste it have at least one dominant allele. Okay, but given that, we do know that 36% of the population is not able to taste PTC. That's important because what that tells us is that the equivalent of q squared in our equations, because q squared represents q times q, and again, that represents the frequency of the homozygous recessive genotypes. So q squared equals 36% or 0.36. Then one of the first things you want to always do is find the value for p and q. So in this case, all we have to do is say, okay, if q squared is 0.36, q equals the square root of 0.36, which equals 0.6. Okay, so we just found the value of q, 0.6. Remember our first equation of p plus q equals 1. In this case, p must be 0.4. So we've just now figured out their expected values of those two alleles, right? 0.6 and 0.4. Now, the question, you may recall, asked for the expected frequency of heterozygotes. Recall this portion right here represents the expected frequency of heterozygotes. So we have P, we have Q. Now we can just say, okay, 2 times 0.4 times 0.6. That gives us a total of 48% or 0.48. So that's our expected or predicted frequency of heterozygotes in this population. Let's go back and take a look at the question itself. So we can see our options here, and we notice that choice B gives us what we're after. So again, this is just a prediction. This is just saying, okay, if all the conditions are met for Hardy-Weinberg equilibrium, we would predict 48% of the individuals are heterozygous. Right? That doesn't say for sure that's what they actually are. That just gives us a prediction. We could then potentially go back and actually do an actual test and sample individuals and see what their genotypes are to see if indeed it matches somewhere close to our prediction of 48%. Okay, so again, that's just one sample question, just to give you a sense of how that can work. So we just went through and said, well, okay, if conditions are met, we have a population that's not evolving. It gives us a kind of a yardstick, a way to measure the extent of evolution that might be occurring in natural populations. So if that's the case, and what are some of the forces that can actually have an impact on the evolution of populations? Well, these are essentially the reverse of the Hardy-Weinberg assumption. So natural selection, of course, can cause changes in allele frequencies. Genetic drift can, gene flow can, mutations can, and mating that is not random can certainly have an influence on the frequency of alleles in population. So let's just explore a few of these in just a little more detail. We already talked about natural selection in a previous video, so we're going to sort of not spend any time on that at this point. So besides natural selection, how else can populations change over time? Well, first of all, genetic drift. What we're talking about here are changes in allele frequencies due to chance, like some random event. So imagine you've got some individual area and you've got a population of butterflies. Maybe it's a mixture of these orange and gray butterflies. And then, a couple of these butterflies fly to new areas and let's say colonize, let's say island somewhere. It's possible that maybe just a few of the orange butterflies go and colonize a new area, in which case then perhaps all of the new populations in that new area would be just orange, or maybe just some of the gray butterflies do the same thing, or you could have a mixture of gray and orange. But the point is you can see how whichever individuals colonize a new area, that's going to have a huge impact, right, on the frequency of alleles in future generations. So we call this the founder effect. Again, it's an example of genetic drift. It's just kind of random. There's nothing saying, orange is any better than gray or vice versa. Neither is selected for here, but it's just that whatever organism happens to colonize a new area, right, that's going to have an impact on future generations and the allele frequencies in those generations. Almost the same idea, but just a slightly different variation on that theme is this idea of a genetic bottleneck. Again, it's an example of genetic drift, but we're talking about a situation where if you have an initial population, in this case, this hypothetical scenario, red represent homozygous dominants, blue represent homozygous recessives, and then purples represent heterozygotes. There could be some random environmental event that occurs. It could be a large fire, it could be a storm, whatever the case might be. It could eventually end up killing off a large portion of your initial population, but a few might survive. And so if we have a scenario like we see here, notice the population to begin with, many more reds, far fewer blues, but just by chance, we could have, after this catastrophic event, we could have a situation like this, where those three genotypes are equally represented. And again, that would have an impact. That would give us a different frequency of alleles, right, in the populations that occur after that catastrophic event. So we say that's an example of a genetic bottleneck. As a real life example, Oliver Sacks wrote a very interesting book called The Island of the Colorblind that tells a story of individuals that live on a very small island in the South Pacific. And they have a very high incidence of extreme colorblindness. And the story goes way back in the 1700s. There was a major storm that ended up killing a lot of the individuals on this island, but a few survived, and the leader apparently had this extreme colorblind allele, and he then passed it on to future generations in his offspring. And so today, again, the individuals that are descendants of that individual who had that allele have this extreme colorblind situation in much higher frequencies than most other regions across the globe. So again, that's just an example of a genetic bottleneck. Okay, let's explore a few of these other mechanisms of evolutionary change. So gene flow. Again, all we're talking about here is individuals mating with other individuals in other populations. So if you have movement of gametes from one population to another, what that tends to do is essentially reduce or minimize any differences in the frequencies of alleles between those two generations. So anytime you have genes moving between generations, again, that can have an impact on the frequency of alleles. And also non-random mating. So, rarely is mating random. There's always some sort of selection going on. So, for example, we can have sexual selection by female choice. Here we see a male peacock doing his best to impress the female and convince her that he's worth mating with, right? But it's sort of her choice. She can choose to mate with him or not, right? So we say, well, that's sexual selection by female choice. Sometimes we can have male-male competition as an example of sexual selection. So these elephant seals, for example, the males tend to battle it out. They can be quite violent. And what you see as a result is the sort of the winner of these battles tends to be the one that mates with the females. And so if you look at populations of these elephant seals, what you see is that just a few of the males end up fathering the vast majority of the offspring. Most of the males, they actually never ever reproduce. So again, there's obviously some selection going on there. Also, another example of non-random mating is inbreeding. And this is actually quite important concept to consider in terms of preserving genetic variation or genetic diversity in populations. But basically, it goes something like this. If you think about inbreeding, we're talking about matings between closely related individuals. An extreme case of that would be those organisms that are bisexual. So many plants, for example, produce both male and female parts. So many plants can self-fertilize. So right, that's the most extreme version of inbreeding. And in those situations, what you see is that that tends to result in an increase in the homozygous genotype, whether it be homozygous dominant, like we see here in red, or homozygous recessive, let's see here in blue. Those genotypes increase in frequency over multiple generations, whereas the heterozygotes, they decrease in frequency. And you can imagine if you're, again, we're self-fertilizing here. So if you happen to be an individual that's homozygous dominant, well, all of your offspring will also be homozygous dominant, right? Same thing with homozygous recessive. And adding to that, if you're heterozygote, well, some of your offspring will be heterozygous, about half of them, but then some, roughly a quarter, will be homozygous dominant, and some would be homozygous recessive. So you see how generation after generation that increases homozygosity in populations. The impact can be fairly substantial. So this idea of inbreeding depression. Often what happens if you have matings between closely related individuals, again, self-fertilization, the extreme version, we have a reduction in fitness. So here we're just looking at plants, some of which were self-fertilized. And then we look at their sort of their reproductive fitness in terms of their offspring, in terms of the flowers that they produce. So when you cross-pollinate these plants, they produce far more flowers over the course of time than those plants that are self-pollinated. So again, this is just one example of inbreeding depression. And then finally, we have to mention mutations. Of course, mutations can occur, they do occur, and this really is the ultimate source of genetic variation. Yes, we can have different frequencies of alleles changing over time, but mutation gives us new alleles with which to work. So this graph here just illustrates some experiments done on E. coli, several populations over the course of many, 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 many generations. And essentially each line represents the number of mutations found in a separate colony. Again, that was propagated over many thousands of generations, just illustrating how mutations can accumulate in populations over time. So that concludes our brief discussion of evolutionary processes.